First speaker will be René Hoet. René will give a presentation and I'll take questions directly after, and then we'll continue with the session 3. So let me first introduce René Hoet. René is, as of the beginning of this year, the Chief Technology Officer of FairJourney Biologics. And I think it's fair to say that René, his whole academic career has been, and biotech career and big pharma career has been in antibodies. His PhD thesis was on autoantibodies. He then moved on to work in Cambridge, where everything with antibodies and phage display was happening at the time. And then he's held major senior jobs in both biotech as well as in big pharma. And it's great to see René come to the basics of it all, the technologies that we're developing to get to the best possible antibodies. Now, for those who don't know René, and there's a famous saying that says, the most scary things are the things you don't know you don't know. And René is the guy that knows the things you don't know. It's that simple. So I'll let him do his presentation and guide us through novel thoughts about antibody design and engineering and what the future looks like. René, the floor is yours.
Thank you very much, Tom, for this very nice introduction. I'm not sure I can answer all the questions here, and I hope you're all enjoying the conference here, the antibody series, and you had a good lunch. And what I would like to do this afternoon, guide you through what our thoughts are on new development of future antibody discovery and optimization, which would try to help to actually enable you to actually make the best therapeutic molecules. So, unlike the talks this morning, where I really enjoyed a lot of biology, this is more on the technology piece I will going to focus. I divided the talk in about 4 parts. The first part I'm going to describe to you what we actually right now offer as technologies for antibody discovery and optimization, and then go a bit more in detail on the mammalian display. You might have read about this already, but I think it's really important to, and I think she will come back to some of the points that were made earlier this morning about developability and biophysical properties of antibodies, how important they are. And then ask the question, so what do we need else? What else do we need for future antibody discovery and optimization? And one of the things people are still trying to improve is actually the antibody library design, having a uniform library which you could actually select out any antibody, and actually I will talk about a modular antibody library which has a potential of 10 to the 27 diversity, and then actually come back to the mammalian display and actually show you a glimpse of what we want to do actually with mammalian display, actually to select multi-specific antibody repertoires. So a bit introduction to FairJourney. We're now actually top leader in top quality customized antibodies programs. We're the largest CRO in Europe. We have a lot of different programs, over 800 different projects we've done, over 200 different customers. From the antibody discovery and optimization work we've done, we know that there are at least 9 antibodies in the clinic. There might be more because we don't get that information from all our customers. And the team right now, which is based in both in Porto as well as in Cambridge, there's over 200 people. And together, both companies actually exist this year, 10 years. So we consider ourselves as a one-shop company for antibody discovery, optimization, but also more and more for production and characterization, including biophysical properties, where we talked about this morning. And also license the different technologies to our customers. And the type of projects we can do in 2 ways. We can do individual projects for if you want to really have an individual problem. But many of our customers who work long time with us, they use what they call an FTE-based model. So they have a number of people working. We have a number of people working for them, and they work on multiple programs and can actually flexibly adapt here. So on top of it, we have actually a large number of scientists. You see scientists that actually day-to-day leading the FAIR journey, with a lot of expertise, over 10 years expertise of doing these antibody programs. But also scientists which have a lot of drug discovery experience, different drug discovery experience, from going from ADCs to bispecifics to antibody discovery. And for instance, also Karen Tillans is actually scientific advisor on preclinical antibody development. So all these advisors, they are for you actually to, if you need a certain problem, to help you solve the problem or to come with a strategy, are available for the programs. So we also try to cover more and more the range from the initial target, the validation of a target, really towards IND. And I will talk today mostly about the first part, the antibody discovery and engineering. And tomorrow, Teresa Barata will talk about the protein science, about the production as well as the biophysical characterization. And then we added actually an additional module of services to our services, and that is actually stable cell line development. And Antonio Barroso will actually explain what we actually offer as a stable cell line development. And the goal is actually in a few years also to include CMC production, so we have the full value chain from target, let's say, validation towards the clinic. So Fair Journey is one company, but we actually have 3 different brands. It's Fair Journey Biologics that have we have actually diverse antibody libraries, naive and immune. I will talk about this a bit more detail. We have display technology, both phage and mammalian display, and we have different type of engineering, which includes humanization, optimization of molecules. That's in the Fair Journey Biologics brand, and we use this for program for our customers. And then there is this Flow 1838 company, which is focused on production of the antibodies. But on top of that, also, we have a lot of assays developed for biophysical characterization and also late-stage development assessment, pre-CMC here. On top of it, we think that all the reagent antibodies that are out in catalogs and so on, the quality of many of those is not, they're not very well characterized, let's phrase it like that. So we are working towards let's say, novel reagent antibodies that are very well characterized and you could use for your research. And finally, stable cell line development, we actually offer basically to all the, let me see how this works. It doesn't work, so okay. All right, so then a stable cell line we also offer, and then tomorrow you'll hear more about that. And then we have the Iontas brands, which is, the activities are ongoing in Cambridge, and there new technologies are developed. The mammalian display is developed in Cambridge, and I will talk about this in a bit more detail. And then customized libraries we make at Iontas. And today I actually will talk about this new Explorer library concept as one of the things which are actually now finalized in Iontas. So we have human naive libraries, and they are already out there for a long time, but they have delivered quite a number of clinical antibody candidates, and we have these antibodies in different formats. Most of our antibody libraries are in Fab format, but we also have single chains because, for instance, CAR T optimization, these single chains are, a better starting point, and we actually can also these phage display selection tools easily transfer to our mammalian display for further optimization. And there's an possibility to license these libraries. These libraries are around 10 to the 10. And then I think that's also an exciting one. We have actually also naive libraries from llamas. They are made of 21 non-immunized llamas, we have them different formats. We have the VHH directly displayed, but we also have them displayed as an FC fusion on the surface of the phage. And also here we can transfer to mammalian display. Libraries typically tend to the 9. We have validated them pretty well on soluble and also more challenging targets. And there's one study here shown, target CXCR4, And there, we, with the help of virus-like particles, which you often actually have to use for GPCRs, we can actually find a quite diverse set of different VHHs, ranging from 0.2 to 80 nanomolar. Some of these VHs very clearly show nice competition in figure 2 with the ligands. And then in figure 3, actually, you can see that the most, let's say, the most interesting candidate, actually, we showed that it actually only recognized CXCR4 and not 4,900 other targets. I think that's another piece which I think most people now automatically include in their analysis before going to the clinic. In the past, people assumed if you have an antibody, it's specific. Nowadays, I think you include this specificity analysis because actually quite a number of antibodies, and from the past I wouldn't know exactly the number, but 20 to 30% of the antibodies which are characterized do recognize a second target. So then, this is the kind of summary slide. So we have a phage display technology where we can display FABs, single chains, and VHHs. Also with the Fc protein there, and we have mammalian display. And the mammalian display, we can actually display complete IgGs and also other formats. I talked about the naive libraries for human, but a lot of our clients actually using immune libraries. Llamas are the most frequently used animal, I would say. We can also combine multiple targets in one immunization to give you actually antibodies to multiple targets at once. That actually saves you time, because you have to make only one library and you can screen then for multiple specificities. But of course also mice, rats, rabbits, and chicken antibody libraries we can make. We do this on a FAB format or single chain format sometimes. And these libraries don't have to be so large. 10 to the 8 is sufficient because this is an immune IgG repertoire, and you don't need such a, let's say, large diversity here. And then today I will talk about more details about this semi-synthetic human explorer library in the second part of my talk. So, of course, not every antibody what you directly get out of a phage display library you can develop. And we particularly for naive libraries, we often have to affinity mature. And this is a summary slide of 14 different antibody programs with different clients. And say on orange you see the parental antibody, and in red you see the affinity matured antibody on the left-hand side. We can do this optimization both with phage display as well as mammalian display. And on the right-hand side you see the fold improvements, which ranges from about 5 to 4,000 fold. So there's an enormous range in how good this affinity maturation works. And this was the focus for a long, long time of the antibody field, focusing on affinities. And, but I want to say, and that's the last sentence of this slide, affinity optimization is much more than affinity maturation. And I rather use the term antibody optimization than affinity maturation. So what else do we do except for improving the affinity? We actually humanize antibodies. So, we make them dehumanize we take the human germline as close as possible to the original structure. And these humanized variants, they are over 90% showing homology with the human genes. And we typically see no loss of affinity. But also, we are very often, and for the design of the mutagenesis, we actually use methods which try to keep the epitope specificity. Functionality is really important. Of the antibody, that's the most important. So you select typically your functionality first. And then with the optimization, we use a rather low number of mutations per CDR, typically about 2 or maximum 3 amino acid mutations per CDR, where we actually improve the affinity. And then sometimes we try to change the specificity. Cross-reactivity is often important, either to cyno or mouse, and that also we can engineer in an optimization. And finally, and that's more or less my switch to also go to mammalian display, CMC, removal of CMC liabilities, undesired specificity, we try to remove in the optimization phase. And that's sort of, going to the next step, mammalian display. People who were a bit early, they maybe already saw the movie from John McCafferty from the Iontas mammalian display. I will want to go a bit more in detail on mammalian display for IgG and then come back to it later for multispecifics. So in the end, what people want if they want a drug, you want to have the perfect, what we call target product profile. The antibody should have the right affinity, functionality, specificity. So those things, I think, when people started to focus first on affinity, now it's all function first. I think all the companies focus first on function. But then something which was underappreciated, I think, is for a long time was that you have to produce large amounts of antibodies which shouldn't aggregate and were highly soluble. And with more and more subcutaneous applications for antibodies, they have to be produced and concentrated at very high concentration. So manufacturability is really an important part. So why, mammalian display? Well, first of all, we can, with the technology which was developed, we can screen 10 millions of clones. Libraries of 10 to the 7 can be made. We gain information on expression as well as affinity. We have the ability to work directly in the final format. This can be IgG, but it can also be another format. And we screen directly in the production type. And that's I think, also an important piece. At the moment, we do mammalian display in HEC293 cells, but also we are moving to be able to do this in CHO cells. And then you actually have your screening of your display system in the same cell type where you make your production cell line. And that could also be a development in the future that you actually move, that we develop a system actually to cross that bridge. And then basically, the challenge for mammalian display compared to, for example, phage display or yeast display for a long time was actually to have one copy of integration per cell, one type of antibody per eukaryotic cell. And then the solution was actually with tail nucleases and nuclease site-directed integration to have one copy of the antibody gene integrated per cell. That's sort of described a bit more in detail here. So there was an intron taken to actually integrate this plasmid, what is shown, part of the plasmid shown is shown below. And there are overlapping regions, left and right, HA regions of about 800 nucleotides with the genome. The plasmid contains a blastincidin gene, which is in-frame cloned. So that need to be expressed to select the clone. And there are two promoters, one for the light chain, PFF, and the CMV promoter for the heavy chain. And this is viewed through transmembrane domain of the PGF receptor for display on the surface. So what happens is actually that with the nucleases, we actually can enhance the stable integration here in the genome. Under selective pressure, you select the cells that have this stably integrated construct. It's about 3 to 5% of the cells do get this effectively in. And then after about 2 weeks, you can actually select these cells with the constructs in flow cytometry for display levels. So how is an example, published example of an affinity maturation of an antibody. It's a PD-1 antibody. On the y-axis, you see the PD-1 binding, and on the x-axis, an anti-Fc binding. And on the right top quadrant, you see actually the binding to PD-1 correlates very well with expression. If you actually would mutate, and we did that, with NNS, and you mutate all the CDR free residues for heavy and light chain, you see that hardly any clone actually binds. That is not the typical method we use for mammalian display, because the diversity of such a library would be too large to be screened with mammalian display. Rather, the mutations are focused on the focused areas. CDR3 of the heavy chain, where 2 mutations were made. You can see the design here at the bottom. So about 10,000 variants made in that. And for the light chain, similar strategy was done, also mutating 2 amino acids per sequence. So in total, there's about 10,000 heavy chain variants and 10,000 light chain variants. So then libraries were made for the VH-only mutant as well as for the VH and VL. libraries are about 10 to the 7 inside, and then selected by the top right quadrant was selected there by flow cytometry. Here's sort of the workflow shown for how such a project would work. So you start, you make the library, you perform fact selections, you rank the affinities and by flow cytometry, and then you do typically a cell-based functional screen where you look at potency and then look at affinity. Here at the bottom you see actually the top candidates which was matured. So the original parental PD-1 antibody is shown in orange. As a reference is Opdivo, nivolumab shown, and then the blue line is actually the improved parental antibody. In potency, it actually shows even a clear, better effect than the nivolumab. And if you look, and that's shown in this slide, to the affinity, you see in this case, and that's not always the case, I would say, but in this case you see it also correlates nice with an improvement in affinity. Parental antibody on the left and the nivolumab on the right, and in the middle you see the improved antibody. So with mammalian display you can optimize and get higher affinity antibodies. And this work has been published a few years ago. But the question, I think the more important question, coming back also to the presentations from Peter Pergier this morning, can you actually select also the antibodies for developability? And it's referring again back to the same paper that Peter mentioned, the paper from Animap by Jane and all, where they characterized 137 clinical candidate antibodies with a lot of assays. And the conclusion here was, the antibodies with biophysical liabilities, they come actually from all the different platforms. And so can we use mammalian display to optimize the developability early in the process, but also more effectively? So this is an example of an antibody where it actually went in clinical trial, bocacuzumab. It actually went to phase 3 clinical trial, and actually it failed, and about 50% of the patients, it's a PSK9 antibody, about 50% of the patients developed immunogenicity here. And in this paper, this antibody was also included, and also there were several red flags for this antibody shown, including a self-interaction and low specificity. Both of those things were described. If we look in our mammalian display system in bocacuzumab and we compare it actually to the humanized variant, and the humanized variant was coming from a mouse hybridoma, and then was improved by phage display from 300 picomolar to 5 picomolar, then actually you can see that bocacuzumab is not displaying that well compared to the humanized variant. So the properties actually got worsened by the affinity improvement. So then, the crystal structure of this was actually described, and then we analyzed, what are the most important residues where probably in the CDRs where it probably is an issue with, and then developed an antibody library on mutating a number of amino acids in the heavy chain and also one in the light chain. And then the top variant, shown here with the red rectangle, was selected and then characterized. So the antibody genes were recovered, they were sequenced, the unique antibodies were expressed, and then biophysical characterization was done of those antibodies. And that's shown here at the bottom. And they are, of course, compared to bocacuzumab, the parental antibody, showing actually also the assays, some of the assays which Peter discussed, like the AC-SINS assay he developed, is shown here. And you can see, for example, that all the improved antibodies, which have an improvement, major improvement in the CDR2, they actually show a better AC-SINS profile. Also, they are much more enriched. You can see the enrichment factors in mammalian display. Most strikingly, I would say, is the polyspecificity difference of this antibody, 215,000, the polyspecificity ELISA, compared to the improved variants. And the two major important pieces are actually reduction of the hydrophobicity, but also to reduce the positive charge, this arginine, which is in CDR2, is actually replaced. That shows a major effect for this antibody. Expression yields have been improved. We go from 70 milligram per liter to 200 milligram per liter, so that's quite a significant improvement. It's actually also compared to another PSK9 antibody at the bottom. You see, it's even behaving better than that one, which is also approved. And also there, we looked at dynamic light scattering, also a measurement for aggregation behavior. You see also there, values are significantly improved. All the antibodies, especially the NEI mutation, are really significantly improving the antibody. So the question is then, if you now look at the immunogenicity, because this antibody was actually 50% of the patient gave an immune response. And there was an experiment done with 10 haploid donors, PBMC donors. So PBMCs were taken from 10 donors. And then in a proliferation, CD4 proliferation assay incubated, and this is, what comes out is that the stimulation index, you look for the 10 donors, and you then can compare bocacuzumab, which is highlighted here in orange, to the best mutant NEE mutation. You can clearly see that, this is a significant improvement compared to bocacuzumab. Bocacuzumab is actually compared, in the same range as alemtuzumab, which is known to be a very, very immunogenic antibody. So you can also, change the stimulation index to, if you take an average of the stimulation and you take the frequency of the donors, then something like responsive index comes out, which is shown on the left. Also here, the same trend is seen. So then actually we looked at, if you look now at the predicted T-cell epitopes, and you look where the changes were made in the antibody with the optimization, we actually saw that there are different spots. So in red, you see the predicted T-cell epitopes, both in the heavy chain and as in the light chain. And in yellow, you see the differences in the now it's going here, yes. In yellow, you see the CDR2 changes, which are made in the optimized antibody. And actually, it's striking because you would expect that there would be some correlation. So apparently, the residues that enhance the solubility and reduce the predicted immunogenicity, I have to say, are distinct from the predicted immunogenic epitopes. And perhaps you could postulate that there might be different immunogenicity, sequence-based immunogenicity, and immunogenicity based on the properties, the aggregation behavior of a molecule. Okay, I want to pause here a bit and switch to what we can do in the future on antibody discovery and optimization. And I won't pretend that I'm going to say in these two topics everything what you can do, because we can do much more. But those two things I think are really important and are on the minds of a lot of people for future drug development. So this is the Explorer module library concept which we designed and at Ionta's site in Cambridge. So the Explorer library, there's sort of on the left-hand side, you see actually the heavy chain and light chain scaffolds we actually designed, and we actually selected a limited number of those scaffolds based on the frequency in humans. We included the most frequently used canonical faults, VH1 and VH3, and also the most frequently used lambda and kappa scaffolds. We also looked in our internal phage display libraries actually if we found those scaffolds often back, because it's not just have to be well expressed in mammalian systems, we also want them to be displayed and expressed well in bacteria and bacteriophages. So that is and then finally, we also analyzed how much they occur in clinically approved therapeutic antibodies. So those were the criteria we used. And then the diversity of the antibodies, we introduced a very designed diversity in CDR1 and 2 of the heavy chains, mimicking the diversity which we see in humans. So some amino acids we left unchanged, also in CDRs, and other ones we changed to the amino acids we see in humans. Similarly, we done for the light chain, and then particularly for the heavy chain CDR, we decided to take this from human donors because the diversity so enormously, varies from 5 to over 25 amino acids that you cannot synthetically design this. And because we take these limited scaffolds and we have a control over the design, we can actually reduce the biophysical liabilities in the design. For example, reduce N-glycosylation, deamidation, etc. we put in the design. So this was taken together, and we made this modular library of, we would like to make this modular library over 10 to the 10 of highly developable antibodies. And finally, because we want to use phage display in per synthesis because you can make very large libraries, and it's a very robust technology, but we want to be able to switch very easily to mammalian display system. So because mammalian display allows us to select for developability. So we have an easy transfer to go to mammalian display for the final antibody format. So this is how the design was done, different modules, module of the CDR1 and 2 of the heavy chain, the CDR3 from the donors, and then for the light chains are similar. For the light chains, we actually did 2 designs for the CDR3. We took them from donors, as well as we synthetically designed. And we will actually evaluate which actually are the best antibodies there, because there's limited diversity in CDR3 of the light chain. So this was all cloned and in these vectors. So this is a bit more illustration of the VH and VL scaffolds we used. We used 2 of the VH1 family and 3 of the VH3 family members, and then 2 lambda and 2 kappa genes for the antibody library, based on this distribution in humans. But in addition, I mentioned also clinical stage antibodies we looked at. So then what we did, we actually made all these sub-libraries of the different antibodies. You can see them here. So the donor CDR3, that's a really important piece to make that diversity as high as possible. So the size is over 10 to the 8, and you can see that if you count all these libraries up, it's over 10 to the 8 of these sub-libraries. Light chain CDR3, less diverse, so there we put 10 to the 5. And then similarly for CDR1 and 2, the different sizes of the library. If you count all these modules up, then you come to a number of 10 to the 27. So you could say, that's not in the library, it's difficult to make in phage, that's true. But in principle, the diversity is there to shuffle around with the initial antibodies you select. So then when we made those libraries, we actually found that there were some errors in the sub-libraries, some frame shifts, 5 to 30%, and we wanted to improve that. And so the libraries, they actually were subcloned with a dummy, what we call just a dummy heavy chain or different light chain for the libraries, and then hyperphage was added. Now, for people not familiar with what is hyperphage is missing the gene 3 protein, which is normally used for assembly in the phage protein. If that phage protein is missing, that means that you need expression of the antibody gene 3 fragment for the bacteria to survive. So you select all the ones that actually express, and then we check the quality of these libraries before and after this hyperphage. And you can see on the left hand it's shown the improvement for the light chain, and on the right hand for the heavy chains. You can see there, especially for the light chain, there's a significant improvement in correct frame antibodies. So the worry a bit is, if you do this type of process, do you not then lose diversity? Because you're manipulating, you're cloning back, are you not losing diversity? And so we did a large check, and all the sub-libraries that we checked 100,000 sequences, and then compared, in this case what you see is the heavy chain CDR3 of the libraries pre and post treatment, and you see actually hardly any differences, identical actually number of CDR3s. On the right hand, on the left hand side for you see the improvement of the libraries. For the heavy chains, it goes from 18 to 10%. For the light chains, it goes from 14 to 3 or 2%. So it's significant improvement. And so we have now made antibody sublibraries here, which are mimicking the human diversity and also have been selected on hyperphage for expression in bacteriophage M13. So then we actually checked, in detail the diversity. So what did we actually obtain? So we compared the design of the antibody library compared what we got in the NGS data. Here is actually just quickly explained how the mutagenesis strategy was. So we put also here a limited number of mutations, only 2 amino acid mutations per CDR in the design. And some amino acids we actually made untouched. For example, the tyrosine in position 2 and the methionine in position 4, they are very conserved and they're stable for stable scaffold, and so we left them intact. And you can see this also matches also what the data we got. And so because we only mutate 2 amino acids per CDR, the number of mutations, so all is shown here. And depending on the position, we put 18, 16, 15 amino acids, depending on what we see in the human diversity. Position 1, for example, you see here the serine in yellow, clearly overrepresented compared to the other 17 amino acids which were included here. And similarly, you see that for the other amino acids, so there is the wild type is overrepresented compared to, but the other amino acids in the design are included. And of course, we left out amino acids which cause biophysical liabilities here. So this is then, the total library made over 10 to the 10, 4.10 to the 10. It's actually not one library, there are 20 sub-libraries made of different scaffolds of the heavy chain and different scaffolds of the light chain. And so quite evenly distributed over the kappa and lambda sides. So about half is kappa and the other half is lambda. So then we did a more detailed analysis, and this is just a few slides to show you that diversity is actually nice what we checked. Similar thing as I said before for CDR1, and I would now take CDR2 as an example. You see that for the VH1 and VH3 family here represented, some positions you have fixed amino acids, other positions you really can have a distribution. It looks like from the slide there is less diversity in here. The reason for that is that we also here put only 2 amino acids per CDR. Since the CDR2 is longer, the number of mutations is also there here less per position. And so those are actually then compared, and they nicely represent what we designed. So then one important piece is the CDR3. People believe that if the CDR3 of a library is not diverse, then you don't get good antibodies. So we thought, how can we now look at the diversity? First of all, we of course do NGS sequencing of all the sub-libraries, so you see a nice distribution, what you would expect, about 13 to 40 amino acids on average size, which is what you find also in humans. But then we also looked at the NGS data in a different way, and in this case, it's just one graph shown. If you look at 90,000 sequences, and you look how many times, let's say, a CDR occurs, you see 80,000 of the 90,000, you see only once occurring the CDR3. And then you see 1,000, actually on the second point, which is almost at the bottom, you see a CDR 2 times occurrence. So it really it's a very steep curve, meaning that the diversity of CDR3 is very, very, very good. And then finally, similar analysis of the light chain. In this case, lambda light chains of the 2 families we included. And here actually in this graph are the synthetic light chain CDR3 is included, but we also have the libraries with the donor-derived CDR3. So conclusion so far, we have made a library of 10 to the 10, which a potential diversity explorer of 10 to the 27, and the results from the NGS quality analysis, they actually show that the actual diversity matches what we designed it in. The next step is actually to validate these library by selecting on some multiple challenging targets, for example, GPCRs. Using the individual libraries because we want to compare the different display levels of these libraries as well as the mix, of course, because that would be the ideal, the one mix of a library. We looked at the NGS outputs, but also look at the affinity and, of course, the biophysical properties of these antibodies. And I think the full power of this library is actually now to use the models. And the way we want to use the models is actually then, once we have the antibody selected by phage display, we want to use individual modules, like, for example, the CDR1-2 module or the light chain module, to shuffle then a certain antibody or repertoire of antibodies to improve the properties, not only affinity, but immediately also the biophysical properties of such a molecule. And we can transfer them to, from the new library to full IgGs, but also to multispecifics. And of course, after validation, we want to use, and hopefully we will soon be able to use this for client-based projects. So that's, we're not there yet. We have to validate it, but so far so good, I think. And this has the potential to be, a very nice tool for many antibodies. So then, going back to mammalian display and multispecific antibody repertoires. This is a slide which I took from a review from Aaron Labine and from Paul Parn, who actually not just in because he had to go to a board meeting. Unfortunately, he apologized not being here now, but his slide. And there's so many different bispecific formats nowadays. And I think Peter Tagier also mentioned it. You guys can't get to what's the final best format. I think, with mammalian display, we can actually probably optimize all of them. The problem, I think, with the multi-specific antibodies is that if you want to select for two functional activity and it's the desire to have the two functional activities really dependent of each other, for example, you want a certain interaction to mimic, then it is really important that you select it in the final format. And developability of those molecules is crucial. So you would like to combine many different antibody combinations, not just one and one, but you want to have hundreds of different antibodies to both targets, including in some of the formats there is linkers included, including potential linkers to be included. All these things you can put in mammalian display and optimize the molecule. So we went through some steps actually to show that this is a nice technology to do. This is a slide showing that if you express VHHs or VHHs FC, we see actually a clear correlation that the good behaving VHHs display much better than the bad behaving VHHs, which is sort of similar as we see with the IgGs. And this is sort of course, a good starting point to make multi-specific antibodies. Then we actually, actually combined some VHHs with single chains in construct. And this is a collaboration project with Argenix. I can't disclose the targets, but this is the type of constructs which were made. So basically, this is an open hole technology where on one side you put a VHH and the other one is a single chain. And particularly with single chains, some behave well and some have problems. So you would like to be able immediately to optimize in the final format. So this is actually showing that these type of molecules you can make very nicely with mammalian display. So these molecules were made in mammalian display, and then with different tags to the molecules, you can detect the different chains. For example, the VHH you can detect with the MIG tag, and the single chain you can detect with the flag tag, but you can also detect with an anti-FC if the total FC is on the surface. So these are the control things we did actually to develop the system, that everything is working well. And then, of course, we looked if you can also select for target binding, simultaneous target binding. And it shows the different controls, the different knob controls, and then different hole controls here for the single chain with target 2 on it, and the VHH with target 1 on it. And you can see, for example, that the single chain, which is attached to the surface of the mammalian cells, if you add the target 2, you can see nicely display on the surface. If you take the VHH as a control, it's a soluble produced protein. you don't see any display on the surface with this with target 1 binding. But if you take the multi-specific constructs, you do see simultaneously target 1 and target 2 binding on the surface. So this is sort of optimized for the targets here. And what we're now doing is actually selecting repertoires of both targets, for the optimal molecule in this program. So in the future, you could actually take, for example, from our new Explorer library, single-chain repertoires, and combining with VHH repertoires from LAMA immunization, but also naive libraries in our mammalian display system, and select for the optimal functional properties. Of course, you could also take any other format. This was just an example, but it shows that you can combine also different formats here in this mammalian display system and select for the for biophysical properties of the molecules. And we can expand to many additional formats, for example, single light chains, biparatopic formats, and CAR T cells. So then I wanted to finish with this slide. Since I didn't talk a lot about functionality, I just don't want to give you the impression I don't think that's important. I think this is the heart of drug discovery, the functionality. I think this is a very nice cartoon review from Paul Carter and Greg Lazar from several years ago, where they highlighted that it's over now with the simple antibodies to HER2 and EGFR and CD20. We now have to go to the high-hanging fruit. To have antibodies which have to pass the blood-brain barrier, where we heard about this this morning, for example, but also to make multi-specific antibodies and have triggers simultaneous function. And I think FairJourney is in a very good position, I think, here to help you to discover and develop these type of high-hanging fruits for the future. 3 short conclusions. We developed this new semi-synthetic human antibody library, which opens up a large diversity, and it's based on a limited number of scaffolds and of highly developable antibodies. Our mammalian display technology allows to select for any antibody repertoire in the desired final therapeutic antibody format. With the Explorer library combined with mammalian display technology, that allows you to have the highly optimal developable molecules for the future. And finally, I would like to thank all the people actually who did this work. So I didn't do that. It is the group in Porto, shown here. The pictures are rather old because these were made actually before the COVID crisis. So we have to come to this conference, which should really make a very nice news slide with all the people in one picture. The people in Porto, but also the people at the Ondas in Cambridge. And I would like to emphasize, John McCafferty, who developed the mammalian display. Also, the Explorer library has been developed at IONTAS in Cambridge. But we're working now together really to explore and develop these technologies further for you and to develop your future antibodies. Thank you.